Hello, my name is Melanie Moses, and I'm from the UNM Departments of Computer Science and Biology, presenting work in collaboration with Stephanie Forrest at Biodesign at Arizona State University, Judy Cannon and the UNM School of Medicine, and Steve Hoffmeyer at Lawrence Berkeley Lab. This is a spatial model of immune response to multifocal viral lung infection. Specifically, we are looking at lung infection in a spatially explicit agent-based model called SIM-Cov. We are asking how virus and immune cell movement through the lung affect infection dynamics and immune clearance. Our hypotheses are that the more dispersed a viral infection is, the larger the viral load and the greater the recruitment of T cells into the lung. We are also based on prior work, expecting that T cells will actually move along physical network structures within the lung in order to get close to sites of infection to kill infected cells. So this is specifically a within-host model of the spatial temporal dynamics of the novel coronavirus within the lung. We explicitly include billions of individual cells and run this on high-performance computing systems. And the model is tested and parameterized with animal and patient data. We have an interdisciplinary team that's worked in computational immunology over the last decades. So our model, called SIM-Cov, will import spatial distribution of COVID-19 infection in the lung from pictures like you see here, CT scans that actually show these white locations where infection has actually taken hold in the lung. Others have used machine learning to convert those into digital images of sites of infection, which we then import into our agent-based model to seed infection at different points in time. We also grow a fractal branching network of the lung airways that are then lined with what are called epithelial cells. These are the cells that get infected and line the airways and allow the virus to propagate. We will also, as I'll show in a moment, include T cell movement patterns within the lung in this model. And we'll output the viral and T cell dynamics throughout a human-sized simulated lung for the full course of infection. Beneath the hood of this model, we are simply looking at the dynamics of the rates at which healthy tissue is infected with virus, releases that virus into the airway and to other cells, and then those cells, infected cells, die. And we also look at dynamics of T cells and how they interact with and kill those infected cells. In order to kill infected cells, T cells have to come into physical contact with them. So this spatially explicit model is very important in understanding T cell dynamics. This video is showing just a quick time slices of how the model progresses as infected cells grow into a larger and larger infected area. T cells then come in and control the infection. We can also look at the same sort of model where we have multiple sites of infection, and the T cells again will come in and control it in the end. But we look at the dynamics of the virus in these two different scenarios where we have the same number of infected cells, but for example, 10 times more dispersion where we have multiple sites of infection, as we see in the novel coronavirus. What we get is nearly 10 times more infected and dead cells in the simulation. We similarly get a huge increase in the number of T cells that are recruited into the lung. This is helpful in controlling the infection because T cells kill infected cells. However, T cells also mediate damage in the lung. And so we are really using this to test the hypothesis that the spatial dispersion of the virus is causing greater immune damage.